Hi, this is George with 573 Medicine, and in this video we're going to be discussing lymphoid histology. So let's get started. A brief overview: the lymphatic system is a group of tissues which are involved in removing waste as well as facilitating immunity. And lymphoid organs can either be primary or lymphoid. So primary lymphoid organs are going to be the bone marrow and the thymus, and the secondary lymphoid organs are going to be everything else, including these diffuse lymph nodes, the spleen, tonsils, as well as structures in the GI tract. And so an easy way to remember is that B and T cells mature in the bone marrow and thymus respectively, so that can help you remember the B and the T for primary, and everything else is going to be a secondary lymphoid structure. In the anterior mediastinum, which is the place between both lungs, we have an organ called the thymus. And this thymus is important for T cell maturation. So now that we understand what the thymus is, let's talk about positive and negative selection. So immature T cells are produced in the bone marrow and express both CD4 and CD8 positive receptors on their surfaces after being produced in the bone marrow. So these immature T cells with both CD4 and CD8 are then going to travel to the thymus, where they will undergo two processes. The first is positive selection, which occurs in the cortex or the shell. And this is where T cells are checked to see whether or not they will bind to self-MHC receptors to make sure that these T cells are functional. And so T cells which cannot bind to self-MHC are non-functional, and these T cells are destroyed. And that's occurring in the cortex, so the outer layer of the thymus, the outer shell of the thymus. And after positive selection, T cells will produce either CD4 or CD8. So they won't express both CD4 and CD8. They'll express either CD8 or CD4. And remember that CD8 positive T cells are going to become cytotoxic T cells, and CD4 positive T cells are going to become helper T cells. The next thing that happens occurs in the thymic medulla, or the middle part of the thymus, the medullary cavity of the thymus, and this is negative selection. And this is where T cells which bind to self-MHC with too large of an affinity are destroyed. So if these T cells bind to self-MHC molecules with too much strength or too tightly, then that's ultimately going to lead to autoimmunity. So in the thymus, negative selection ensures that these T cells which bind self-MHC too tightly are destroyed, so you don't have autoimmunity. Then these mature CD4 or CD8 positive T cells are able to leave the thymus after negative selection. So this is the histology of the thymus. You can see that it is surrounded by a fibrous capsule, and there's the cortex, which is the outer shell, which is where positive selection occurs. And then we have the medullary canal or cavity, which is where negative selection occurs. In the cortex, we're going to have thymic epithelial cells. In the medulla, this low-hanging fruit to easily help you identify that it's the thymus, we have these Hassel's corpuscles. The function is currently not well understood, but it's an important landmark for identifying that you are in the thymus, specifically the thymic medulla. So this is just a brief close-up of the cortex. So these arrows are actually pointing to thymic epithelial cells. And this is the thymic medulla with a zoom in of the Hassel's corpuscles. This is an adult thymus, and this picture just shows you that the thymus, compared to the child's thymus, is going to atrophy with age and be replaced with adipose tissue instead of those abundant lymphocytes. So now that we have zoomed in on the thymus and the bone marrow was discussed in a previous video, let's zoom in on secondary lymphoid organs, or everything else. This is a close-up of a lymph node. So each of these little dots are going to be lymph nodes, part of the lymphatic system. And lymph nodes have a cortex, which is also known as the outer cortex, as well as an inner cortex, which is known as a paracortex. So the outer cortex, or just the cortex, receives lymph via afferent vessels. And in the outer cortex, there are primary follicles, which contain dormant B cells, and secondary follicles, which contain active B cells. And the germinal center, as well as the mantle zone, are part of the secondary follicles, which are part of the outer cortex. The paracortex, or the inner cortex, contains high endothelial venules, which are vessels which allow T cells to enter the paracortex. So the paracortex, you are going to find these T cells. In the medullary canal, or the medulla, we have medullary cords, which contain plasma cells, as well as medullary sinuses, which contain macrophages and reticular cells, as well as are responsible in efferent lymphatic communication, so outside of the lymph node. This is just a zoom up of an H&E of a lymph node, and feel free to look at this picture and try to figure out what structures are inside of each of the parts of the lymph node that are labeled in this picture. So the next organ we're going to be talking about is the spleen. So the spleen is an organ which is located in the left upper quadrant, of the abdomen, which plays an important role in immunity. There are two types of pulp. There's the red pulp and the white pulp. So the red pulp is going to filter red blood cells and metabolize hemoglobin into bilirubin. And these contain sinusoids with elongated endothelial cells. So sinusoids means similar to a sinus, but not quite a sinus. And there are elongated endothelial cells. So these are going to be special types of linings of capillaries that almost resemble sinuses because they're so open to allow old red blood cells to be filtered and metabolized in the spleen. You'll also have the splenic chords, or the cords of Billroth, in the red pulp. The white pulp contains lymphocytes and monocytes, which are going to be important in degrading extracellular pathogens which have been opsonized by antibodies. The parts of the white pulp include the follicle, which contains B cells, the periarterial lymphatic sheath, or PALS, which contains T cells, and the marginal zone, which surrounds the periarterial lymphatic sheath and contains macrophages. Something which may not be as obvious to you is that red pulp is going to have red blood cells, and white pulp is going to be having lymphocytes and monocytes, which are types of leukocytes, which are also known as white blood cells, if that helps at all. So this is just a great overview of the anatomy and histology of the spleen. So we can see that it's encapsulated, as well as having this trabeculated meshwork. We can see that there are these sinusoid capillaries, or capillaries which are like sinuses that allow red blood cells to enter and be metabolized by the spleen. And we have these aggregates of lymphatic tissue, as well as germinal centers, in the white pulp. Additionally, we have the central artery, and the central artery is surrounded by that periarterial lymphatic sheath, or PALS, this is going to be something that you can just kind of look at later on and appreciate these structures. But the spleen is honestly kind of challenging, so you might have to look at this a couple of times. This is another zoom in of an H&E of the spleen. So we have the fibrous capsule, the red pulp, as well as these aggregations of lymphoid tissue with a central artery, and that's going to be our white pulp. So this is a zoom in of red pulp, and we can see the sinusoids with actual erythrocytes, so these red cells without nuclei. And we can see the splenic chords, or the cords of Billroth, as well as endothelial cells which are lining these capillaries. This is a zoom up of white pulp surrounded by red pulp, and this vessel right here is going to be the central artery, which is surrounded by the periarterial lymphatic sheath. So that's it for this video. If you liked it, feel free to